This is John, and John is sick. John is currently experiencing different symptoms such as a cough, fever, shortness of breath, difficulties breathing, and the loss of taste and smell. John finds himself in a dilemma, unsure about if he should go to work or not. He thinks about his sister, who is studying biomedical sciences, and decides to give her a call, and hopes to get some useful advice. After explaining and reviewing his situation, his sister encourages him to get tested. John decides to go to the nearest test station. After arriving at the test station, a nasopharyngeal swab is conducted, collecting nasal secretion from the back of the nose. The swab is then placed in a tube that contains a buffer solution. The buffer solution serves as a transfer medium, removing all substances such as viral components and proteins from the swab. The sample is now ready to be analyzed in the lab. The first step is the transfer of the sample solution into a compatible tube for the following procedure. The tube is then placed in a heat block for several minutes. Heating the sample causes a destruction of the native virus, releasing the ribonucleic acid and other proteins. The single-stranded RNA contains all the genetic information. In order to amplify the viral genetic material, the obtained RNA needs to be transferred into a double-stranded complementary DNA with the help of the enzyme reverse transcriptase. In the graph, the three main steps necessary for a polymerase chain reaction are demonstrated. These include the denaturation of DNA, the annealing of primers, and then the extension of primers. The PCR process is fully automated and conducted in a thermocycler, which takes around 4 to 5 hours depending on the amount of cycles. Coming back to the newly obtained cDNA, the first step of the PCR is the denaturation, separating the double-stranded cDNA into two single strands. This step takes place at 95 degrees Celsius. Once the cDNA is single-stranded, the next step, the annealing, can take place. The goal is to equip each single strand with a complementary strand. To reach this goal, primers are attached to the single strands. Primers are short, specific nucleotide sequences complementary to the viral cDNA. Fluorescent reporters attach to a specific part of the single strands for later detection. The third and final step, the extension, starts with the binding of the Taq polymerase. The Taq polymerase is a thermostable enzyme that synthesizes the complementary strand. As soon as the Taq polymerase reaches a fluorescent reporter, the binding fluorescent signal is detached. The fluorescent reporter consists of a fluorescent signal and a quencher. Quenchers are substances that decrease the fluorescent intensity without destroying it. The enzyme polymerase cleavage cuts the bondage between the reporter and the fluorescent signal. The results are determined by the amplification of the fluorescent signal. The threshold is a diagnostic cutoff parameter. It is the point where positive and negative results are classified and differentiated. Every amplified fluorescent signal above the threshold is considered as a positive test result, and everything below with no amplification is a negative test result. After a couple of days, John received a letter where he was declared as SARS-CoV-2 positive. John is feeling scared, anxious, lonely, and isn't sure about how to proceed, especially because his symptoms have already improved. He decides to call his sister and discuss the situation with her. His sister encourages him to stay at home and always wear a mask once he leaves the house. She also sent him a video showing a simulation of the consequences and how the coronavirus spreads without people following the required restrictions. The purple dot in the center shows a COVID-positive person, such as John, if he wouldn't follow the rules. As John gets in contact with more people who are initially healthy, he starts infecting them, resulting in a chain reaction that more and more people get infected and spread the virus without knowing it. As shown in green, some people are recovered after being infected. After watching the simulation, he realized what a true hero in our current society looks like. Being a hero means staying at home, wearing a mask, keeping distance, and limiting contacts. Stay safe and keep healthy.